The human body is not a sterile battlefield, it is a living ecosystem. From the moment we are born, trillions of microorganisms settle on our skin, in our gut, and along our mucosal surfaces. They are not accidental invaders. Many are permanent residents, shaping our physiology in ways we are only beginning to understand. Not all microbes come with harmful intentions. In fact, most exist in balance with the host. This balance defines the host-microbe relationship, a dynamic interaction that can range from cooperation to confrontation. In mutualism, both host and microbe benefit. The microbiota synthesize vitamins, assist in digestion, and train the immune system. In return, they receive nutrients and a stable environment. It is a partnership refined through evolution. In commensalism, the microbe benefits while the host remains unaffected. These organisms quietly occupy space, neither helping nor harming. They are silent tenants in a crowded microbial city. But when the relationship shifts to parasitism, the story changes. The microbe benefits at the expense of the host. Nutrients are stolen, tissues are damaged, and disease begins to unfold. At birth, colonization begins almost immediately. Resident microbiota, formerly known as the normal flora, establish long-term communities, while transient microbiota pass through temporarily. Over time, this microbial population becomes an integral part of our defense system. Microbiota are not passive bystanders. They compete for nutrients, occupy attachment sites, and produce substances toxic to pathogens. This phenomenon, known as colonization resistance, prevents harmful microbes from gaining a foothold. Colonization alone does not equal disease. Microbes may be present on body surfaces without causing harm. Infection begins only when organisms invade tissues, multiply, and trigger an inflammatory response. Once infection occurs, outcomes vary. Some infections remain subclinical, with no noticeable symptoms. Others progress to overt infectious disease, marked by fever, systemic inflammation, and local manifestations according to the site of infection. Whether a disease develops depends on two forces: microbial virulence and host immunity. Pathogenicity denotes the ability to cause disease, whereas virulence quantifies the degree of that ability. Some microbes are mild opportunists, others are aggressive primary pathogens. Primary pathogens can infect healthy individuals without assistance. Opportunistic pathogens, however, wait patiently before striking when immune defenses are weakened, barriers are breached, or chronic illness is present. Pathogenic bacteria have many virulence factors, the molecular weapons they carry. Adhesins allow attachment, invasins permit tissue penetration, toxins damage cells, capsules protect against immune destruction. Let's walk through how bacterial infection is fully established and how the virulence arsenal is exploited. The first step of infection is adherence. Without attachment, microbes are swept away by mucus, saliva, or urine. Structures such as fimbriae act like grappling hooks, anchoring bacteria firmly to host cells. Biofilms create sticky communities resistant to both immunity and antibiotics. Infection advances when bacteria move beyond surface colonization. Using specialized proteins known as invasins, pathogens bind to receptors on non-phagocytic cells, manipulating the host cell into engulfing them. What looks like forced entry is actually molecular deception. The bacterium turns the host's own machinery against itself, gaining entry into a protected intracellular niche. Once inside, multiplication begins. Binary fission resumes, numbers rise, the quiet colonizer becomes an expanding threat. Iron is essential for bacterial metabolism, yet the human body hides it carefully. Host proteins such as transferrin and lactoferrin lock iron away, limiting microbial access. This defense is known as nutritional immunity, but pathogens respond with siderophores, high-affinity iron-binding molecules that steal iron from host storage proteins. In infection, survival often depends on who wins this microscopic tug-of-war. As immune cells mobilize, pathogens activate survival strategies. Capsules form slippery shields, preventing phagocytosis. Leukocidins destroy white blood cells directly. Coagulase generates fibrin barriers, cloaking bacteria within protective fortresses. Immune evasion is not passive resistance, it is active counterattack. When gram-negative bacteria lyse, they release lipopolysaccharide, or LPS, from their outer membrane. The toxic component, lipid A, triggers an aggressive immune response. Unlike exotoxins, endotoxin is not secreted. It is released upon bacterial death, making even successful antibiotic therapy capable of temporarily worsening inflammation. Endotoxin activates macrophages and induces massive cytokine release, including tumor necrosis factor alpha, interleukin-1, and interleukin-6. The consequences are systemic: fever, inflammation, vasodilation, and hypotension. In severe cases, this cascade progresses to septic shock, characterized by dangerously low blood pressure, organ failure, and high mortality. A molecule measured in micrograms can destabilize the entire circulatory system. Unlike endotoxins, exotoxins are actively secreted proteins. They are highly potent, highly specific, and often lethal in very small amounts. Because they are proteins, they are antigenic, meaning they can stimulate antibody production and can be converted into toxoids for vaccines. Exotoxins do not require bacterial death to act. They are weapons deployed during life. Many exotoxins follow the AB model. The B subunit binds to a specific receptor on the host cell, the lock and key step. The A subunit enters the cytoplasm and disrupts cellular metabolism. Protein synthesis may be inhibited. Second messenger systems may be altered. Cell signaling may collapse. It is a targeted biochemical sabotage. Other exotoxins work by forming pores in cell membranes or enzymatically degrade phospholipids. The result is cell lysis. In red blood cells, this becomes hemolysis. This category of toxin causes immediate structural damage rather than metabolic interference. Some bacterial toxins act as superantigens, taking a different approach. Instead of targeting cells directly, they overstimulate the immune system. By non-specifically bridging antigen-presenting cells and T cells, they activate large numbers of T cells simultaneously. The result is explosive cytokine release, leading to hypotension, fever, and shock. Unlike precise AB toxins, superantigens create chaos by overwhelming immune regulation. The immune system is powerful, sometimes too powerful. In its effort to eliminate pathogens, it releases cytokines, activates complement, and recruits inflammatory cells. But this response can cause collateral damage. Tissue destruction, edema, and organ dysfunction may result not only from the microbe, but from the host's own defense mechanisms. Disease is often the price of an intense immune response. Understanding host-microbe interaction transforms infection from a random event into a logical sequence. Colonization precedes invasion. Invasion triggers inflammation. Virulence competes with immunity. Outcome reflects balance. Microbiology is, therefore, not merely the study of microbes. It is the study of relationships, of coexistence, conflict, and survival.